Experimental guidelines for intracellular cytokine detection. Cytokine stimulation, transport inhibition, cell fixation and permeabilization and flow cytometry antibody needed in the experiment. Self-prepared reagents including cell staining buffer, deionized water and absolute ethyl alcohol. Sample preparation. Prepare human peripheral blood single cell. Collect white membrane cells. Add appropriate amount of culture medium to wash cells. Centrifuge at 300 times gravity for 5 minutes. Discard supernatant. Add 2 ml culture medium to centrifuge tube. Resuspend cells. Count cells. Cytokine stimulation and transport inhibition. Adjust cell concentration to 1 to 2 million cells per milliliter. Blow and mix fully. Transfer cells to cell culture dish. Gently shake the cell culture dish to make the cell suspension evenly cover the surface. Prepare 500 times cell stimulation mix solution. Add 100 microliters cell stimulation mix solvent to each tube of cell stimulation mix. Thoroughly blow and mix fully. Prepare 1000 times protein transport inhibitor mix. Add 50 microliters anhydrous ethanol into each tube of protein transport inhibitor mix. Thoroughly blow and mix fully. Add 500 times cell stimulation mix at a rate of 2 microliters per milliliter of cells. Blow gently and mix softly. Place cells in an incubator at 37 degrees Celsius and 5% carbon dioxide and incubate cells for half an hour to an hour. Remove the cell culture dish after incubation. Add 1000 times protein transport inhibitor mix at the dosage of 1 microliter per milliliter cell suspension. Blow and mix fully. Place cells in an incubator at 37 degrees Celsius and 5% carbon dioxide. Incubate for 5 to 16 hours. When incubation is complete, remove the cell culture dish. Collect cell suspension. Rinse with appropriate medium and collect the rinse solution. Cell surface staining. Centrifuge at 300 times gravity for 5 minutes. Discard supernatant. Add appropriate amount of cell staining buffer to the cells. Blow and resuspend cells. Take 100 microliters cells into the centrifuge tube. According to the antibody instructions used, add flow cytometry antibodies to stain the cell surface marker. Incubate cells at 4 degrees Celsius for 30 minutes in the dark. Remove cells after incubation is complete. Add 1.8 milliliters cell staining buffer to each tube. Resuspend cells. Centrifuge at 300 times gravity for 5 minutes. Discard supernatant. Add 200 microliters cell staining buffer to each tube and resuspend cells. Cell fixation and permeabilization. Add 200 microliters fixation buffer and mix fully. Incubate at room temperature for 30 to 60 minutes in the dark. Dilute permeabilization buffer to permeabilization working solution. Fully mix the solution. Add 1 milliliter permeabilization working solution to each tube of cells. Centrifuge at 600 times gravity for 5 minutes. Discard supernatant. Add 100 ml 1 times permeabilization working solution to each tube and resuspend cells. Intracellular cytokine staining. Add the corresponding intracellular flow cytometry antibodies according to the antibody instructions and mix well. Incubate at room temperature for 30 minutes in the dark. Add 1.8 ml cell staining buffer to each tube. Resuspend cells. Centrifuge 600 times gravity for 5 minutes. Discard supernatant. Detection. Add 200 microliters cell staining buffer to each tube and resuspend cells. Detection.